So yeah, carrying on with where we sort of got to looking at gene mutations in cancer. So we've looked at those types of gene mutations that we can have. So we've said that bases can we can go through those additions or substitutions, or we have those point changes or those inversions. And we'll have a quick recap of those at the end, the different types of mutations we can have. But we're going to look at how they can link to cancer, and actually also how epigenetics can link to cancer as well. So cancer is a disease that can affect animals and people of all ages, and there's lots of different types of cancers, but they all involve basically that uncontrolled growth, and all have, and it's uncontrolled growth, and that's uncontrolled growth often due to mitosis. So we're going to look at how the cancers can occur, basically, to start off with. So different types of genetic mutations and something called a mutagenic agent, which is what we talked about before. So we're saying we can have different agents and they can basically increase the rate of mutations occurring. We're also going to look at something called cystic fibrosis as well, really quickly. So quick recap of what a gene mutation is. As we've said before, a gene mutation is any change either in the amount or in the structure of your DNA from what it was originally. Here, they're going to use two types of mutations because we can basically group them, but obviously we have more than just those two, so I will go through them all again. But we can say a gene or a point mutation. So this is where we've gone in and we've changed a base sequence of a gene. So in the base sequence of a gene, there's a change, and that can cause a change in the polypeptide chain. And this is caused by errors that occur during DNA replication. So when DNA is being replicated during mitosis, a base has changed in the sequence of a gene, and that can then obviously cause one of them what we called a frame shift, and that's where obviously it changes afterwards. So then all our polypeptides can change after. Therefore, the protein that's created isn't necessarily the right one. It might not be functioning, or it could function and it could be harmful. Its effects, or the fact that we're not making the right protein anymore, can be harmful to the body. So it's sometimes not that we're creating something harmful, it's the fact that we're not creating something that should be there, then has a bad effect on the body. And these occur during DNA replication. Or we can have a chromosome mutation. And this is when we have a change of a number or the structures of our chromosomes. And this occurs during cell division. So obviously that's going to actually be where we've got those chromosomes aligning along the center of our cell. When those centromeres attach and they get pulled apart, those sister chromatids, during cell division, that something has gone wrong there. So there are two big groups of mutations. So all those mutations I talked about before, so like substitutions, deletions, inversions, point changes, duplications, they are all DNA replication mutations. And then we can have these chromosome mutations as well. So, quick look at some of them. These are just some of the mutations that we can have. Quick recap. So substitutions, sometimes called nonsense. So this is type of mutation that results in a stop codon being coded for. This will prevent the rest of the polypeptide chain being produced. So basically with substitutions, we can get different types of substitutions. I'm not saying that we can't get deletions and stuff, but basically what happens here is, why is that not going again? So annoying. Basically, the mutation results in a stop codon being coded for where it shouldn't be. And so then no more of that polypeptide chain will get coded for. So rather than arginine, aspartame carrying on, this stop codon comes in and it just stops. So it's not making any sense. It's making nonsense. We can have substitutions that we call missense. And these types of mutations result in a different amino acid being coded for than what we wanted. And then this can affect the structure and function of our polypeptide chain. So these are the typical ones that we sort of talk about. We've changed one base, change that polypeptide, so then it can change everything else. So nonsense substitutions, which brings in a stop codon. Substitution that can change one amino acid, which can then affect that whole protein because of its folding later on. We can also have silent substitutions. These type of mutations result in our same amino acid being coded for. Remember, even though our base has changed, and that is because our genetic code is degenerate. So there is no change in our polypeptide chain, even though the base has been substituted for the wrong base. And then we can have a frame shift deletion. So the type of mutation alters the reading frame of the gene. So everything downstream of the mutation. So it will cause a different sequence of amino acids to be coded for. So basically we've got rid of something and then everything else afterwards has then changed. So it's one of those frame shift ones. So deletions, insertions will also cause frame shift, inversions as well. So quick recap now of mutagenic agents. So the natural spontaneous mutation rate, because we do have natural spontaneous mutation rates in our body, it's typically around 1 or 2 mutations per 100,000 genes per generation. Okay, so that is natural. However, our mutation rates can be increased by things called mutagenic agents. So things like UV radiation and harmful chemicals like carcinogens, these increase our mutagenic rates. So these disrupt the structure of the DNA and they can interfere with transcription. So there we go, interfering with transcription. It's not just DNA replication, this is interfering now with our transcription. So we're now looking at those epigenetic changes as well. So mutagenic agents aren't necessarily a change of the structure of the DNA. It can be, so they can come in and permanently change the structure of your DNA, or they can interfere with transcription. So there could be an epigenetic effect. So it's going to maybe be methylated or acetylated the DNA. So is it basically how coiled tightly around the histones are they, or are our methyl groups in the right place? So mutations provide this genetic variation that we need for evolution. However, as we know, we can have harmful effects. So one disease that you do need to know a little bit about is cystic fibrosis. It's recessive inherited disease. So what do we mean by that? Recessive, meaning it's not dominant, which means we need 2 of the same alleles for it. Inherited means it's passed on from generation to generation. So if we were to draw a really basic Punnett square for it, and let's use this for cystic fibrosis. These are the parents, both parents are homozygotes. So let's actually do a Punnett square, that is not helpful. So C is our dominant, capital C, that's our recessive. So both parents are homozygotes. They're both carriers, so you can only get cystic fibrosis either from someone that has cystic fibrosis, or both parents must be carriers, otherwise you can't pass it on. But you could be a carrier yourself. So if you get both of the dominant genes, which means you don't have it, you'll be homozygous without it. Here you're going to be heterozygous, so you're going to be a carrier, and that'll be no disease. Here you're going to be heterozygous and a carrier, and then here you would have cystic fibrosis. So you've got a 1 in 4 chance. So obviously, if you had a parent who had cystic fibrosis, but then a parent who was who didn't have it and didn't carry it, and now I can't show it very well, you would always be a carrier, but you wouldn't ever be a sufferer. And then raise all them. If you had a parent who had cystic fibrosis and a parent who was a carrier, you could get it. So again, you'd have a disease, you'd have a 50-50 chance to get the disease, and then you would also always be a carrier. Last example, if you had a parent who was a carrier and a parent who didn't have it at all. You could have a 1 in 4 chance of being a carrier, and then the other times you wouldn't carry it and you wouldn't have the disease. So my random going off on that. So cystic fibrosis is caused by a number of different point mutations in the CFTR gene. Lots of diseases aren't actually caused by one mutation, it's by quite a lot of different ones. The CFTR gene is all about mucus production, and it codes for transmembrane protein that acts as an ion pump. So basically it's a protein. So the CFTR gene codes for a protein that's called the CFTR protein, and it acts as an ion pump. And this ion pump is to do with sodium. And basically it controls how much salt is coming in and out. That salt control and concentration is what basically affects how much mucus is basically given into our body produces or doesn't produce. And basically this fault in the ion pump causes people with cystic fibrosis to produce too much mucus. So our CFTR gene is found on chromosome 7, and it codes for 1,480 amino acids. And there are over 1,000 known mutations which can affect the function of the CFTR gene in different ways. So that same basically CFTR gene itself codes for 1,480 amino acids. So it's long, like we were saying the other day, genes are long chains of amino acids. In 70% of cases, cystic fibrosis is caused by triplet deletion. So we've deleted a whole triplet codon. So that results in the complete removal of amino acid from the polypeptide chain that we're producing. So let's just close that now. And let's get on to, why has it done that? Part about the cancers. So basically, like I said at the beginning, cancer is a group of diseases that are caused by abnormal cells dividing uncontrollably. This forms an undifferentiated cell mass. So if we think back, differentiated cells are our specialized cells. Undifferentiated cells, we often refer to as stem cells, but we can also have them in the form of tumors. So this forms undifferentiated cell mass called a tumor that can destroy healthy tissues. Cancer will start in one part of the body, but our tumor can break loose, it can break off, and then it can spread to other tissues and organs. So once it's in a different part of the body, it metastasizes and it grows somewhere else. And it's not a single disease, and there's no single cause or cure for cancer. So every year there's around 320,000 people diagnosed with cancer, and approximately 156,000 people die of it in the UK. Now, our incidence and mortality rate of cancers can differ depending on the type of cancer. So if we have a look at the 4 most common cancers for men and women, we've got obviously men and women in different graphs. So for men, the highest is highest incident rate. So most common cancers are the ones that occur the most. So ones that occur the most in men are prostate cancer, followed by lung cancer, colorectal, and then bladder. And for women, it's breast cancer, lung cancer, colorectal cancer, and uterine cancer. Now, if we look at the incidence rate, that's a dark green area. Prostate cancer has the highest incidence rate, but on that graph, it hasn't got the highest mortality rate. Its mortality rate is just over 20%. Now, it might be the second highest on that graph, but not 20%, sorry, 20,000 per 100,000 men, 20 of them would die from it. However, if you look at the amounts, if you're comparing them to basically how many men are diagnosed with it and then how many people die from it, there's actually a huge difference if you look in the percentages. And so actually there's a big difference there. Whereas when you look at something like lung cancer, 60 men per 100,000 are diagnosed with lung cancer, and actually of those 60 you're looking at 50 of them dying, so only 10 would survive. So the mortality rate for prostate cancer is a lot lower. Whereas if you look at bladder cancer, it's a lot less common. It's about 19 men. But the mortality rate, again, is quite low. It's probably about 2, 4 maybe. But you don't have as many men being diagnosed with it. And it's very similar if we look at women with breast cancer. So you've got about 122,000 women being diagnosed with breast cancer per 100,000 women, but about a 22% mortality rate. So that is a difference. Whereas, and then you look at lung cancer, there's not as much as big a difference, or colorectal. Uterine, there's quite a big difference. Now, you might think, well, why do more women survive from colorectal than men? Because that does happen, but we also have less women being diagnosed with it than men, so that could be why their mortality rate is also lower. So yeah, so cancers, incident rates and mortality rates change, and mutations that affect these individual cells are what are going to cause them. So we have 2 types of tumors. It's really good for us to cover this again to make sure that we know the difference between the 2. It was something covered at GCSE, but I always like to go through it again. So benign tumors. A benign tumor is a mass of cells that has not spread into neighboring tissue. So it's controlled, it's in one set area. The location of that tumor is restricted to that singular part of the body, and it's in one type of tissue only. So the example here shows a colonic polyp. So that's a polyp in the colon, and that's going to be caused by abnormal growth of the epithelial cells in the intestinal lining. We class benign tumors as non-cancerous. Okay, so cancer, you'll often find people interchangeably using the term cancer and tumor, and they're not the same thing. A tumor is massive cells. Okay, so tumors can develop for years in your body, and they might not have any obvious symptoms, and they can be quite large. But it's really important to remember not all tumors are cancerous. So we have the 2 types, benign or malignant, and benign are not cancerous. They generally grow slower than malignant tumors, and they're often covered in fibrous tissue as well. And that fibrous tissue is really important because it stops them invading other types of tissue. So for example, this colonic polyp, it's just of the epithelial cells in the intestinal lining, and as you can see, it's got fibrous tissue over it. It's not going to go and invade any other type of tissue. They're often harmless in terms of cancer, but we do often remove them for other medical reasons. So for example, they could cause blockages, or they can put pressure on other organs. So we generally remove them because of their physical presence rather than what they could become. So you might have a tumor growing. So lots of brain tumors are benign, but sometimes people have to have them removed because they can place pressure on, like, for example, your eye, or it can place pressure onto other parts of your brain, which then can stop other parts of your brain functioning and working properly, but it's not the risk of them metastasizing. In a similar way, this one in the colon, you would want to remove it because it could cause a blockage in your intestines. So we wouldn't want that. There is also the chance that some benign tumours can eventually turn malignant, so we do like to remove them for that reason as well. So they don't invade surrounding tissues because they are not cancerous. However, they have potential to develop into cancers, and that's because they already have that uncontrolled cell growth characteristic to them. So that characteristic can lead itself to becoming cancerous. So the most common method to remove benign tumours is surgery, and basically the affected tissue is just cut away. Then we have malignant tumours, and these are known as cancer. So when people talk about cancers, these should be classed as malignant tumours. These have the ability to invade other tissue, and they can destroy tissue, and they grow very, very rapidly. And these tumours are uncontrollably dividing, and the cell groups that may spread or metastasize to other parts of the body. They don't always, but they can. And if we leave them, they often will. So for example, here we go. We've got a CT scan of 3 brain tumours, and these are metastasized from breast cancer tissue. So some of that cancer tissue is broken off from the breast and then it's traveled to the brain and it's metastasized in the brain. See the 3 brain tumours here, here, and here. So we can treat, so basically how it would have got there. So because they're growing rapidly and they can invade and destroy double tissue, these cells can actually break off. The tumours can break off and they can spread around to other parts of the body, and they can get there in 2 ways, either via the blood system or via the lymphatic system. So the lymphatic system being how like our hormones and other stuff move around our body. So malignant tumours can be treated in a couple of different ways, and that depends on a few things, basically what type of cancer it is, where in the body it is, and what stage that cancer is. So it's often a combination of surgery and then other treatments. So some cancers can be treated with drugs to kill the fast-growing cells, and that's chemotherapy. We can also use radiation therapy, which uses ionizing radiation to damage the DNA of the cancer tissues to kill the abnormal cells. It's quite interesting because obviously we've said radiation is a mutagenic agent, but actually it's really good for getting rid of cancers. So, cell division. Cancers occur because of uncontrolled cell growth. So mutations that are going to occur in individual cells after fertilization, and this is really important, but we'll talk about this is after fertilization. So this is in adulthood. We call these acquired mutations. What's happened? So yeah, we call these ones acquired mutations. So we've got them throughout our life. We were not born with them. So if these mutations occur in genes that control the rate of cell division, mitosis, they can cause uncontrolled cell division. And basically, if our cell divides uncontrollably, the result is a tumor. And that's our mass of abnormal cells. And the tumors that invade and destroy are called cancers. And obviously, they're the two. We're going to look at the 2 types of genes that can control this in a minute. So basically, our cells need to divide over time. They're going to divide to make sure that dead or damaged cells are replaced. And we can control that by 2 gene classes. So we can have proto-oncogenes, and these stimulate cell division, okay? And then you can have tumor suppressor genes, and these slow or inhibit cell division, which is important. So in a normal cell, the activities of these 2 gene classes are really, really balanced. We don't want too much of one, not enough of the other. However, if the expression of these genes change, this is where problems can start to arise. So let's look at a proto-oncogene, for example. So the proto-oncogene increases cell division. If we increase the effect of a proto-oncogene, and that can be caused by mutation. So for example, if a point mutation occurs in a proto-oncogene, it can form an oncogene. So we get rid of the proto. And what this does is this can stimulate excessive cell division and it can lead to a tumor. So basically, we get rid of the word proto once it becomes mutated. So when a proto-oncogene becomes mutated, we get rid of the proto and it just becomes an oncogene. And that's once it's mutated. When these proto-oncogenes function normally, they stimulate cell division. And they do this by producing proteins that make cells divide. If a mutation occurs, so then if a mutation occurs in our proto-oncogene and becomes an oncogene, the gene can then become really, really overactive. So then it stimulates excessive cell division and it causes the cells basically to divide completely uncontrollably. And so then the rate of division increases and a tumor is formed. If we then look at a tumor suppressor gene, these can then be inactivated. So depending on the mutations that occur. So if a point mutation occurs that can activate it, what happens is basically tumor suppressor genes work when they function normally by slowing down cell division. It prevents cell division from going too fast. They produce proteins that actually stop cells from dividing, or they cause the cells to self-destruct. So, and that's apoptosis. So tumor suppressor genes, how they work normally, so this is how they would normally work, is they produce proteins that either stop cells dividing or they cause a cell to self-destruct, which is apoptosis. Destruct, yeah. Called apoptosis. So if a cell has got a mutation, apoptosis can occur. If a cell is too old, too damaged, apoptosis can occur because sometimes we don't actually want to replicate that cell. That cell might not actually be a great cell to replicate, so we just get rid of it. Or if we've only just made a new one of that cell, the tumor suppressor gene is going to pump out those proteins that's going to stop that cell from dividing for a while, because it's like, hang on, we've just made a new skin cell. We don't need to make a new skin cell anytime soon. Like, chill out. We don't need to make that many. So that's the general idea of them. However, if, like I said, one of these point mutations occur in a tumor suppressor gene, those proteins that stop cells from dividing or cause apoptosis aren't produced at all. So this one, we get basically, if there's a mutation, more proteins made. And then in our tumor suppressor gene, our proteins that should be made to stop cells from dividing aren't made. So then our cells divide uncontrollably, the rate of division increases, therefore we get a tumor. And that's the basics of it. DNA methylation. I mentioned this the other day. So before I do that, I just actually want to go through something about tumor cells before we do DNA methylation. I want to put in with something randomly. Just going to make sure I'm sharing my screen to the right place. While that's loading, I want to just go on about tumor cells, how they look and function differently to a normal cell. So tumor cells differ from normal cells in a couple of different ways. And it's quite good to know these because you can often get asked these as a question. So the first way they differ is they have an irregular shape. You might get given an image and asked, how do you know this image is, we've got a tumor cell present. So like, for example, if you had like squamous epithelial cells, let's just have a go at drawing some. It'll be interesting. They might all, oh, I was not expecting that to happen. They might all look a bit like this. You've got your really badly drawn nucleus inside, and then suddenly you might start getting something like this occurring. So we've gone from something that's really routine, really regular, sort of square shape, to then these weird shaped parts. I mean, you might get these square ones back over here. And some really strange square ones. And so this part would be normal, this part would be normal, and for normal, and then this section would be a tumor. So irregular shape, straight away, that's a warning that that's one way of knowing that it's working differently, and it differs. So yeah, irregular shape. Second thing, I didn't draw that part on very well, so I'm going to draw them on here. Oh, they're moving. Why are they moving? I don't know what's happening anymore. Two, what is happening? Let's see if I can get this to write a bit more. bit more normally. I'm not quite sure what's happening. What I was messing about with is I was trying to show, right, let me close that and open it again, because that is just going to make me cross. What I was trying to show you was that the nucleus can get bigger and darker. So once my whiteboard's open again, here we go. Let's try this again. I'm going to share the screen with you. I know it's not up at the minute. Let's try this again. Okay, so we've got a regular shape. Oh, it's staying in the same place. The nucleus of tumor cells, the nucleus is darker when you're looking at it under a microscope, and it is larger than in normal cells, and larger. Sometimes as well, they might have more than one nucleus. Stick one here. Sometimes more than what? The third thing, they don't produce all the proteins needed to function properly. So not all proteins produced. The fourth difference between tumor cells and normal cells is they have different antigens on their surface. That's really actually quite useful. When people are looking into new medications, new ways to try and combat cancers, if we've got different antigens on their surfaces, maybe there's a way we can just target those cancers to get rid of them, rather than, for example, in chemotherapy, affecting all the cells in those areas. So that's where like personalized medicine starts coming into it. We start looking at those antigens on them. 5, they don't respond to growth regulating processes. We know that that's how they've ended up forming, but it is good to know. So they don't respond to growth regulation processes. And then the last one, 6, they divide more frequently. So more frequent division, more frequent cell division, and they are the 6 key ways of telling the difference between the two. So methylation, epigenetics, we were talking about DNA methylation the other week, links to cancer. So let's have a quick look how it links to cancer. So DNA methylation. Basically, DNA methylation, we've said, is the adding of that methyl group. Methylation is just adding a methyl group onto something. Methylation of DNA is important because it's a method of regulating gene expression that we said. Basically, it controls whether or not a gene is transcribed and translated into a protein. So when methylation is happening normally, like we said, it's really, really important in the body. If we've got too much methylation or too little, that's when we've got issues. And so tumors can be caused by abnormal methylation of those cancer-related genes, such as the tumor suppressor gene and the proto-oncogene. So as we can see, the methyl groups have been added to the DNA, and the methyl groups present basically prevents enzymes involved in transcription from binding to the DNA at that site. If the DNA is over-methylated, it can block certain genes completely, which just means that those enzymes can't attach, and then those genes can't be translated and then transcribed. So methylation switches off genes, so it inhibits their expression. So if a gene is then demethylated, it's switched on. We've removed the methyl groups, and then it can be translated. So that's epigenetics, as we said. So hypermethylation is too much methylation occurring on the DNA. So DNA methylation, like we said, completely normal, and it affects different genes throughout your life. However, some environmental factors, such as smoking, increase the risk of abnormal DNA methylation patterns. So for one example, tumor suppressor genes are usually switched on to slow down cell division. So we don't want them to be methylated. We've got them switched on, we want them to be transcribed. If parts of your tumor suppressor gene, now it doesn't have to be the whole tumor suppressor gene, because if you remember how long genes are, but even if just part of it becomes heavily methylated, that gene then will become deactivated. It's switched off, it's not going to be transcribed, it no longer inhibits cell division. So basically, if this tumor suppressor gene becomes hypermethylated, we cannot transcribe the gene. So if we don't transcribe the gene, the proteins aren't going to be produced, and those proteins are what slow down the cell division. So this will then mean that our cell can divide uncontrollably by mitosis and tumors can develop. Oppositely, we can have hypomethylation, and that can be a negative side effect as well. So proto-oncogenes, which are those ones that increase the division of cells, don't know what was happening there, is usually suppressed by DNA methylation. If we demethylate those proto-oncogenes, the gene is then permanently activated, and then cell division is stimulated. So if we get rid of that methyl groups, it causes them, as we said before, remember, we don't just call them proto-oncogenes now, it then acts as an oncogene. It then increases the production of the proteins that encourage cell division, and this stimulates the cells to divide uncontrollably, which causes then the formation of tumors. So estrogen, we talked about estrogen the other week. I'm going to look at it again. So certain breast cancers grow in response to estrogen, and estrogen is a sex hormone. And basically, these cancer cells are going to have estrogen receptors. And increased exposure to estrogen over a long period of time is thought to increase a woman's chances of developing breast cancer. So increased exposure may be the result of a few things. So it could occur due to starting menstruation earlier than usual, or starting menopause later than most women. It could also be a result of taking estrogen-containing drugs such as HRT or the combined contraceptive pill. So full reasons behind why we might have increased estrogen isn't fully understood. There's a few theories. So these cancer cells have estrogen receptors, and what happens is when estrogen binds to these receptors, they send a signal to the cell to stimulate cell division. The higher the body's estrogen concentration, the more these cancers will grow. And this type of breast cancer can be treated by hormone therapy, where the body's estrogen production is suppressed. So actually it's treated very differently to lots of others. So basically the idea about this, I'm just going to swap screens a second, is a load of chemistry coming up there. We think there's three ways that the estrogen works and can contribute to some of the breast cancers. We think that the first way is that basically estrogen can stimulate breast cells to divide and replicate. So estrogen stimulates breast cell division. So if you've got more estrogen present, then you're going to have more cell divisions occurring. And so if you've got more cell divisions occurring, naturally, this is going to increase the chance. So stimulates breast cell division. So we've got this increase in cell division, which is naturally going to increase the chance of mutations. Therefore, these chances of mutations increase the chance of them becoming cancerous. The second way we think estrogen might cause cancer is the ability to stimulate division could also mean that if the cells do become cancerous, their rapid replication could be further assisted by estrogen. So actually, so the cancerous cells are dividing more rapidly due to estrogen. And then finally, we think actually estrogen might be able to put mutations directly into the DNA. It's a little bit unknown. So let's quickly switch back. What we were looking at here. Nope. Genetic risk factors. So mutations, we can have mutations in specific genes. So one you've probably heard of is the BRCA1 and BRCA2 genes, and they're thought to be associated with breast cancer. And so this graph shows the incidence of women developing breast cancer below the age of 70. So we've got total population, but if you have BRCA1 mutation, you've got a lot higher chance of having cancer than the general population, similar for BRCA2. So BRCA1 and BRCA2 are tumor suppressor genes, and these produce those proteins that help to repair damaged DNA. So the mutations in them basically stop them from working properly, which means those proteins aren't released, which means you have a higher chance of developing breast cancer. So this is also looking at environmental risks now. So this graph, we're looking at breast cancer incidence, age, and alcohol consumption. So it's showing that the women who consumed the highest amount of alcohol are more likely to have breast cancer, but also as you age, the incidence rate of breast cancer increases as well. Could we call that direct evidence that alcohol causes breast cancer? No, we can't. We can say there's a correlation, but we can't actually say it's a direct evidence because actually those women who haven't drank alcohol, they're also finding breast cancer in their body. So we can't say, no, it is definitely that. What environmental factors other than alcohol might affect breast cancer? Well, you could say it's to do with lifestyle. So you could say that maybe people who drink alcohol maybe aren't as healthy, maybe they're not sleeping as well, so that could affect it. We might have other chemicals that people put into their bodies, sunlight. There's lots of things that could affect it in those situations, as well as age in that situation. So to finish off, I just want to have a quick look at some exam questions. It's going to come up with some maths. Don't worry, we're not doing maths. It's some GCSE maths. Just open from my work earlier. Oh, it's still showing the maths questions. Right, so I think we did this question the other day. Explain how structured DNA is related to its functions. Yeah, we looked at that. Here we go. So I want to talk through this question now with you. So I'm going to sort of talk through it and then have a go at answering it for you and sort of work through it that way. I'm just going to increase the light because I can't see that anymore. So scientists investigate 3 genes. So we've got gene C, gene D, and gene E. And these genes are involved in controlling cell division. So what they did is they studied the effect of mutations in these genes on the risk of developing lung cancer. So the scientists analysed gene C, D, and E from healthy people, and they looked at people who got lung cancer. So if a person had a normal allele for the gene, so we've just got a bit of information here on these sorts of questions. So on these sorts of questions, we put in some information that lets us know how we're going to actually read and understand this table. So if a person has a normal allele for a gene, we use the symbol N. And if a person had 2 mutant alleles, then we use the symbol M. So they use this data to calculate the risk of developing lung cancer for people with different combinations of N and M alleles of the genes. A risk of 1 indicates no increased risk, and the following table shows the scientist's results. So these are our people that they are looking at. So if you have got, let's look at the first one. Gene C, no mutation. Gene D, gene E, your risk of developing lung cancer was 1. So that's our no increased risk. So this obviously is our control group. So that's always good to make sure we know we've got our control group and where it is. That's our control group. So mutation in gene C, risk is increased by 0.3. Mutation in gene E, risk increases by 0.78, and then in gene D, increased by 0.45. So what does the data suggest about the relative importance of the mutant alleles of C, D, and E increasing the risk of developing lung cancer? And asks you to explain your answer. Now let's have a look how many marks this is. So it's a 3-mark question. So we want basically 3 points about this, and it asks you to talk about C, D, and E. It's really clear. What does data suggest about the relative importance of mutant alleles of genes C, D, and E on increasing the risk? And again, they've highlighted it there for you, so you should know the risk of developing lung cancer, and explain your answer. So let's not do that because that is not helpful. Let's scroll this up and have a go at answering this. So which one has a high risk? Well, the highest risk is gene E, so that would get us a point. So we could say gene E. Gene E produces the highest risk. I would always say include the numbers in there as well. If you've given it us, it's helpful. I would always include it. So gene E, well, I'm saying gene E, but a mutation in gene E would be better. So a mutation in gene E produces the highest risk. I'm going to include that, which is 1.78. Next mark, D produces the next highest risk. I would include the number again, which is 1.45. And then C has the lowest risk. Mutation C is the lowest risk at 1.3. That is all you need. 3 marks, and basically the 3 marks are just for putting them in order. You could literally probably summarize it in one sentence, say mutation in gene E produces the highest, D is the next, followed by C. A sentence for 3 marks. So if we look at the next part of the question, chemotherapy is the use of a drug to treat cancer. The drug kills dividing cells, and the figure below shows a number of, and this is important, let's have a look at what it's telling us, the number of healthy cells and cancer cells in the blood of a patient receiving chemotherapy. The arrows labelled F to I show when the drug was given to the patient. So F to I, so this is when the patient was given then until then. So what this question asks us to do, okay, let's zoom this out so I can actually see the whole thing, is calculate the rate at which healthy cells were killed between days 42 and 46. Let's see if we can do this on here then. So what would be important in this situation then is to find days 42 and days 46. I have made that way too big now. I'm having a nightmare with this today at making sure it's a sensible size. Right. We are now blurred. This is not helpful to anybody. Oh no. Give me a second. It's decided to close itself because it wasn't happy at how many times I have pressed that. Now it's decided to come back. So calculate the rate at which healthy cells are killed between days 42 and 46. So I want to find days 42 and 46. Let's count how many days we've got. Well, probably in twos, aren't we? 2, 4, 6, 8, 10. Yeah, 50. So days 42. We are line 42. So that's day 42. I presume, yeah, is H, which makes sense. So day 42 is H. Oh, that did not underline it, never mind. Day 42 and day 46. So 2, 4, 6, and that is day 46. So day 42, what are we looking at? 1,200, 1,400. And then day 46. 4, 5, 6, 7, 2, 4, 6, 8. So 800 there. So let's try and do some mad quick maths. I would normally do this in my head. However, when I do this in my head, under pressure, I always do it wrong. And I've done it wrong already because I wrote a number that I can't even read. So that should be, what should that be? Oh no, so that would be so 4. Oh yeah, so that's be 570 maybe, 570. So 830. So what are we doing? So calculate the rate at which healthy cells were killed between days 42 and 46. So yeah, so we'd got how many, about 830 killed, so 1,400. So that's 830. How many days was that between 42 and 46? So 43, 42, 43, 44, 45, 46, 5 days divided by 5. So I think I've read this wrong slightly, but basically you want to work out the difference between this point and this point, take them away from each other. I got roughly 830, but I think I've done it wrong counting. Then how many days it was. So we've got day 42, day 43, day 44, day 45, day 46. So divide it by 5 days. And then that would give you the rate at which they're killed. I've got 166, which means I've read it wrong because I know, because I double checked earlier, the answer should be 180. And this is what happens when you try and read a graph on a computer. So that shows you a good example of why you need to use a ruler to find your lines on your graph. Yeah, because I've gone too low there. I think that would probably be more like 575, 590. But anyway, 580. But yeah, that's a general idea. So you're going to find out your difference between H and that point there, take them away from each other. Then you're going to divide that number, whatever it was. So I had 830, for example, by how many days it was. And that'll give you how many cells killed per unit volume of blood per day. So last little bit, describe similarities and differences in the response of healthy cells and cancer cells for drugs between times F and G. So let's find F and G. So here's F, here's G. Well, what similarities and differences? Okay, well, our similarities are, there's a similar pattern. F goes down at F, healthy cells go down, cancerous cells go down. Stay the same at this point. Number of cells stays the same for the same length of time. Yeah. Then they increase again. So how many marks is this? 3 marks. So our similarities are going to be same pattern. Decrease. Stay the same for the same amount of time. Stay the same. That's for the same amount of time. And then increase. So we've got that same pattern. Also ask you to talk about differences. So let's have a look at some differences. It's going to be to do with per unit volume of blood, to be quite honest. So if we have a look, we could say there's a greater, faster decrease in number of healthy cells as compared to cancerous cells. So you can say greater decrease in healthy cells to cancerous cells. Let's have a look at other differences. We can also say about the increase as well. We've got a greater increase in the number of healthy cells than the cancerous cells at that point too. So